for your presentation. I'd now like to welcome Henry Sebesta to the stage to begin the next presentation. Welcome, Henry. The floor is yours whenever you're ready.
Okay. Hi, everyone. My name is Henry Sebesta, Product and Business Development Manager for Chromatid. I've been with Chromatid for a little over 4 years, and I've gotten the chance to do some of the product development work for a few of the assays I'm going to be discussing today. So I thank all of you for joining me, and I thank scientists for hosting this. Today, I'll be walking through how Chromatid's proprietary cytogenetic analytical platforms, directional genomic hybridization, and pinpoint FISH can be used as in vitro research tools to assess the genomic impact of gene and cell therapies for oncology. First, a bit of context for those who may not be familiar with us. Chromatid is a cytogenetic analysis company based just outside of Boulder, Colorado. At the moment, we're a team of about 30, but we've been rapidly expanding over the past year or so, finishing up a B round of financing at the end of 2021. We were initially a spin-off of a radiation biology lab at Colorado State University, funded by NASA as a way to monitor the effects of ionizing deep space radiation on their astronauts. We still do some of that work, but our primary focus these days is on helping gene and cell therapy developers bolster their regulatory data filings with unique single-cell analysis that combines the innovative aspects of our technology with the validated standards of regulatory requirements. You can see here from a couple of studies with our first customers in the space, Intellia Therapeutics and Editas Medicines, where we help them identify and track translocation rates in their treated cells. We do this by using a technique I've already alluded to called directional genomic hybridization. This technique is similar to FISH, but uses our proprietary bioinformatic design and cell prep methods to selectively strip daughter strands during cell replication. This leaves a single unidirectional chromatid on either side of the centromere in metaphase chromosomes that, when we hybridize our single-stranded oligo probes to, the result is a hide-based assay that has orders of magnitude better resolution than most FISH probes, and also captures every class of genomic structural rearrangement, including translocations, copy number variants, inversions, and sister chromatid exchanges. There are two iterations of this technique that we differentiate primarily by the amount of genomic coverage of the probed area. DGH Insight on the left here is our localized version of the assay with a resolution down to about 2 to 3 kb. It's mostly used to track edit sites or characterize transgene integration events associated with editing or viral delivery. DGH Screen on the right is our unbiased approach where we paint every chromosome with probes so we can identify down to a specific chromosomal arm where rearrangement events are happening throughout the genome. Screen can be used for a variety of applications ranging from genomic stability assessments for cell line characterization to identifying off-target or unintended genomic effects of any therapeutic modality. You can see a couple of real-world examples of Insight and Screen here. On the left, what you're looking at are Insight probes in one metaphase spread of a dual edit system in hex cells. Those circles there are highlighting the green and red probes spanning 2 edit sites on 3 homologs. There are 2 examples of a normal edit site where the red and green probes are on the same side, and an example of an inversion here where part of the red probed region between the edit sites flipped over to the opposite chromosome. On the right, we have our prototypical karyogram for Screen. This is meant to demonstrate Screen's ability to detect various rearrangements throughout the cell and how, using different labeling patterns and chromosomal morphology, we can identify which arms of the chromosomes are occurring. Before we get into some data, I wanted to highlight some of the concerns around current popular analytical methods. The World Health Organization and the National Institute for Biological Standards and Control put together a collaborative study to try and establish a standard for lentiviral integration analysis. What they found was that there was some variability between the 31 labs that participated and between the 3 PCR-based methods used when trying to determine lentiviral copy numbers per cell. I've included an excerpt from the study here, which I think summarizes the overall issue nicely. The bottom line is that while PCR data is critical, there are limitations around amplification biases, reaction conditions, and data interpretation. So the global gene therapy community is still on the lookout for analytical methods that can help bridge the gap. Here's an outline of a study we did that's representative of how we propose to shine some light on this issue. You can see here ideograms of the study we set up with a collaborator that was using an unidentified viral vector to deliver an integrational transgene to what they thought was a targeted site on chromosome 22. For this study, we made probes for their target site in green, their integrant in yellow, and we added paints on chromosomes 1, 2, and 3 in the background as proxies for monitoring genome-wide off-target effects. Here's an example of a cell from their control human dermal fibroblast line without their integrant. You can see the green probes there bracketing their target integration site. but we wouldn't expect to see any integration here, so all looks normal. What we found in the rest of their iPSC samples, however, was not normal and was a bit unexpected for both of us. We found that after looking at about 600 cells across their samples, only 6% of those cells had integrations that were only at the target site. A good portion, about 40%, had both on and off-target integrations, and almost half of their cells had no on-target integration whatsoever. You can imagine they were pretty surprised, as they were expecting a much cleaner system. So we went on to analyze another cohort of about 200 cells, gathering the same data but with a slight twist. To this cohort, we added a control probe on chromosome 8 to try and help us quantify the copy number at each integration site. The control probe on chromosome 8 was of a known genomic size and fluorescence intensity, and by using that and the known genomics of our insert probe, we were able to calibrate our other signals against it to give an estimate for the number of inserts at each integration site. In addition to even more drastic off-target data observed in the second cohort, we were also able to glean some pretty interesting copy number data. You can see the number of integration events on the y-axis here and the integrational copy number on the x-axis. This data indicated that the vast majority of these inserts, both on and off target, were being integrated as only one copy, although we did see a fair amount of variance, including 9 integrations with over 5 copies. So, to summarize, we were able to provide our collaborator with not only a cell-by-cell distribution of the number of integration events, but an event-by-event analysis of the number of inserts at each integration site. Shifting gears a bit here, I'll quickly walk us through another case study to demonstrate potential applications for another iteration of our technology, DGH screen. This study was performed with the NIST Genome Editing Consortium, and we are analyzing the genome in a bottle progenitor cell line provided by the Coriell Institute. In addition to being the most sequenced cell line in the world, as they say, Coriell had already performed G-banding analysis and found a potential inversion on 3q. So the goal of the study was to use DGH to confirm previously observed rearrangements and to see if we could find anything that might have been missed. You can see here a good example of one metaphase spread from these cells that shows a variety of rearrangements were present throughout the genome. As you can see, many of these were large structural rearrangements like translocations and inversions that were difficult to detect using the previously described deep sequencing methods. Without getting into a detailed breakout of the prevalence of each rearrangement, we were able to confirm the suspected inversion on chromosome 3, and we also found quite a few new inversions and unexpected rearrangement complexity. Here are a few examples of some of those newly discovered inversions and the recurring complexity associated with chromosome 16 on the left there. In addition to that multi-radial structure that we observed in 4% of cells, there were other hallmarks of instability, including decondensed or elongated centromeres that we were able to confirm with standard karyotyping methods. Here's an example image from our G-banding validation on the same cells. Much of the instability and rearrangement data on chromosome 16 matched our DGH results, as you can see with that elongated centromere right there in the middle. But as expected, there were many rearrangements, including the previously known and newly discovered inversions that we weren't able to see with G-banding, which kind of highlights the power of screen's kilobase level resolution over the megabase level resolution of the current regulatory karyotyping standard, G-banding. So with that, we've walked through a couple high-level applications of how our DGH platform, but I want to briefly touch on the last iteration of our technology, pinpoint fish. The oligos we use for our DGH probes can also be used as extremely high-resolution, single-stranded fish probes, which we can then ship to you as kits or run in our lab as a custom assay service. Our bioinformatic design enables researchers to run 2 to 3 kb resolution fish assays as opposed to the approximate 100 kb lower limit of back-based fish, which can be run in your lab using standard fluorescence microscopy equipment. We also recently launched our first standard fish kits, which include a variety of sub-step and sub-tilo probes, as well as our TP53 and SEP17 gene kit, all of which range from about $15 to $18 per assay. You can learn more about our fish kits through our website or by reaching out to sales@chromative.com. So we've gotten a chance to cover a variety of applications for our technology, including our methods for edit site tracking and integrant profiling with DGH Insight, unbiased whole genome stability and rearrangement assessments with DGH Screen, and our ultra-high resolution fish kits and assays. This workflow provided here is an example of one of our service projects, but as I've mentioned, we're also happy to ship you out a kit and let you run these assays from the comfort of your own lab. At the moment, many of our customers are combining Insight, Screen, and G-banding for the ultimate cytogenetic analytical suite that provides targeted, unbiased, and standard validation for their regulatory filings. So far, we've contributed data to 4 different filings from 4 different companies, with a 5th on the way, and we hope we can continue to increase that number with a few of you. If you're interested or you think you have a potentially relevant project, we'd be happy to discuss it with you. Please feel free to reach out to the contact info here or my personal contact info provided here. Thanks again to everyone for your attendance. I really appreciate your time and attention. And thank you for scientists for giving us the opportunity to present.
Thank you, Henry. That concludes the presentation portion of this event. So we'll now move into the Q&A. And as a reminder, if you have a question, you can submit it using the Ask a Question panel that's shaking on the screen here. Okay, let's jump in. It looks like we have quite a few questions here. This is really exciting. Oh, excuse me. Let me just start to bring some up here and we'll get going. Okay, great. So it looks like we have a wonderful first question. So is BioIVT able to detect mutations using liquid biopsy?
Hi, Megan. Yes, we are actually in the validation process of obtaining cell-free DNA using different isolation methods to validate looking at mutations for liquid biopsy. So that's in development as we speak.
Excellent, thank you. Another question for Dr. Bunka. A really interesting talk, thank you. You showed some data about testing the specificity of these vehicles against cancer versus healthy cell lines, but obviously there is a lot of heterogeneity of cell types and protein receptor expression in the human body, particularly when it comes to the hypothesis-free targeting. How do you ensure there aren't off-target effects in other diverse cell types outside of more homogeneous cell lines?
Yeah, that's a very good question. It actually comes down to how you design your selection approach. So every aptamer selection program is different. And we actually spend a lot of time talking to our clients about the sort of off-target effects that you might experience in a real-world situation. So we try and build in as many of those off-target cell lines as possible. So not just targeting cancer versus healthy in, let's say, the pancreas, but you might want to look at other sort of epithelial cells, for example, if you were going to be injecting it. So it's all about getting that cell line right. And we can use multiple cell lines if we want to. Now, there is an addition that we can do as part of the selection process is what's referred to as in vivo selection, where we actually take an animal model, for example, and put the whole aptamer library into that model. So in that situation, the aptamers are going to be exposed to every sort of tissue type as they circulate around the body. And then when we recover the tumor, we'll obviously be recovering the aptamers that have survived that circulation around the body and made it to that tumor. It's a bit more involved. It's a more lengthy process, but it's something that we're very interested in doing. I hope that answers the question.
Thank you. This one's for Kathy. What QC acceptance criteria are used to determine if the DNA and RNA extracted from an FFPE block are suitable for NGS screening?
That's a great question. We actually go through a number of different validations. So we use quantification using qubit for DNA high sensitivity as well as RNA broad range assays. We use the RIN number as well as the DIN functional range for DNA and the base pair sizing range, all from Agilent Technologies. And then we also do DV200 analysis. However, we like to make sure our clients are aware that this data is really dependent upon the isolation method and the technique that is used, which both of those can have an impact on the RNA fragmentation. So it's very important to look at that.
Thank you. This next one is for Henry. What type of cells do you require for DGH analysis?
Yeah, so DGH can be done on any cells that you can get metaphase spread out of. The DGH portion does require actively dividing cells, and so some of the most common cells we work with these days are iPSCs, T cells, NK cells, and then we accept live or cryopreserved samples You can use your culture methods, or we can help you develop.
And how many cells do you need to look at for DGH analysis?
Well, the number of cells we look at is going to vary depending on the prevalence of the event you're looking for. And so the less frequently you expect to see the event, the more cells we're going to need to count for statistical power. So if you're looking for edit site rearrangements, for example, we'll probably have to count more cells than if we were screening clones. But we typically fall in the 200 to 600 cells counted per sample range for edit and integrant tracking, and then maybe in the 20 to 100 range for the whole genome analysis.
Thank you. The next one, it looks like, is for David. Can aptamer binders be used in cancer applications other than therapeutics?
Yeah, absolutely. So we're actually using our aptamer binders in all sorts of applications. So not only therapeutics, but diagnostics, research tools. Essentially, anywhere that you could use a binder, you can use an aptamer. So you might, for example, in your studies, use flow cytometry. So we can generate an aptamer binder that's specific to a cell type, pop a fluorophore of choice on there, and then use that as a flow cytometry reagent or a FACS reagent, so you can actually sort and recover your cells. And as I said, you can also use them potentially as diagnostics as well. So there's a lot of examples out in the literature where people have conjugated all sorts of tags, including radioisotopes, for example, to allow the aptamers to be used as tracer molecules to look at sort of tissue localization, cancer sites, that sort of thing. So yeah, all sorts of applications. It's not just therapeutics.
And do you select for aptamer binders with endosomal escape properties?
Very good question. It's not something we specifically select for, but as you could see in some of the examples that I showed in the presentation, there is evidence that the aptamers are binding to cells, getting internalized, and then escaping those endosomes and making it into the nucleus. So, we don't specifically select for that, but there is evidence that it happens anyway.
Excellent. And one more for you. If you select for cell phenotype rather than a specific biomarker, how can you discover the specific receptor that the aptamer targets?
Yeah, again, that's a very good question. You're quite right. Any selections that go through our hypothesis-free approach, at some point you're going to need to work out what that biomarker actually is. So this is actually becoming more routine practice. So you can actually use a binder molecule to pull out a biomarker, so attach it to a solid support like a magnetic bead, for example, and use that to bind to the surface of a cell, so stick to its receptor of interest, lyse those cells, and then pull that biomarker out and send it for example, tryptic digest and mass spectrometry. So there are methods out there for identifying those biomarkers once you have a binder.
Wonderful, thank you. Henry, it looks like we have a couple more questions for you. The first one is, can you only work with human cells?
No, we can work with anything with a sequenced genome that we can design probes against, including research models like mouse, pig, or non-human primates.
And then for the DGH analysis, do you only run that as a service?
No, actually, like we touched on a little bit at the end there, we also sell our custom and standard probes as kits. And so any of these assays can be run in your lab. You'll just need a fluorescence microscope with the appropriate filters. Our cell prep method and hybridization protocols are pretty simple, but we also offer on-site or team support for those who would feel more comfortable with us walking through.
Thank you. And then we have a couple questions for Kathy. Is BioIBT able to detect mutations using liquid biopsy?
Yeah, so we're validating that process right now with cell-free DNA, and we have some initial promising results.
Excellent. And if you've used BioIBT for NGS mutation data screening, are you able to have access to the raw data files?
Yes, actually, we share that as a matter of principle. So we have the ability to share the FASTQ, the SAM, the BAM, and the VCF using cloud-based storage solutions based on the client's needs.
Excellent. And it looks like this next question is for David. What is the size limit of molecules that can be conjugated to aptamers?
That's a very good question. I'm not actually sure the answer to that. I know that we've conjugated on site here everything from small molecules, which are down to about 1.5 kilodaltons, I think was the smallest ones that we've conjugated, all the way up to double-stranded siRNAs. So they are in the region of sort of 30-ish kilodaltons. But we've seen examples where people have conjugated whole liposomes, for example, or even viruses. So you can conjugate things that are up to megadaltons if you need to.
Excellent. And this one looks like it's for you as well. Do aptamers induce immune responses?
Oh, that's a very good question. That's one I get asked all the time, actually. They can, but most often they don't. So it really depends on the sequence of the aptamer and the modifications that are held in its backbone. So it's well known, for example, that particularly GC-rich sequences can stimulate toll-like receptors, so you get complement activation, for example. But for the most part, if that happens with your aptamer, you can actually engineer those sequences out, so reduce the GC content of your aptamer to limit that effect. But for the most part, they don't stimulate the immune response. It's only a very few sequences that do.
And another question for you. What are the PK or PD properties of aptamers in vivo? This is reminiscent of the nanoparticle story where targeting looked great, but in practice, they were taken up by liver macrophages. Any data on systemic delivery of aptamers in vivo?
That's a very good question as well. Yeah, so it's very true that aptamers, like any other nucleic acid or a lot of other drugs, they do accumulate in the liver, in the kidneys, mainly because that's their route out of the body. But there are a lot of studies that have been done with subcutaneous and IV injections of aptamers. And what they quite often show is that the aptamers circulate around the body, localize in the tissue of interest, so where they've been designed to go, and then you see a pool of them in the liver and kidney as they're excreted. So you do see tissue localization as well as accumulation in the liver and the kidney.
Excellent. Thank you so much. Another question, does your technology require cell clones?
Is that for aptamer, sorry?
You know what? That's a good question that you have. I'm not sure who that question was for. Okay. Well, with that, little bit of a confusing question. Thanks so much for joining us for today's presentation, and a big thanks to Aptamer Group, BioIVT, and Chromatid for sponsoring this cutting edge conversation. We now invite you to take a moment to share your feedback on the survey, and we'd really appreciate your input. If you still have a question, or if we didn't ask your question to the wrong person, it's not too late. Feel free to send it in now, and we will forward it along to the speakers following the event. If you're interested in learning more about Aptamer Group, BioIVT, and Chromatid, we invite you to check out the resources panel. And if you're interested in seeing what's coming up next in the Cutting Edge Conversations series, you can click the register button on the slide to find out more. In closing, we hope you enjoyed this Scientist.com webinar, and we'll see you next time.